Hello friends, today we are going to understand the early experiment in the unit photosynthesis in the higher plants. So let's proceed. The very first experiment in this was explained by the scientist Jan Ingenhouse. What did he do? He took the green plant, that is the aquatic green plant, and then he covered with the transparent glass so that the sunlight can pass through this green plant. Now, what did he observe? He observed the formation of bubble near the green part, and he gave this name as the oxygen. Now, later, he performed this experiment in the dark condition. But in the dark condition, there was no bubble formation and no photosynthesis took place, obviously, as no bubble formation took place. So he concluded that the sunlight is necessary for photosynthesis. Later, the another experiment was explained by the scientist Julius von Sach. What did he do? He provided the evidence for the production of glucose when the plant grew. And this glucose is usually stored in the form of starch. Later studied showed that the green substance in the plant is nothing but this substance is chlorophyll, and this chlorophyll is located in the specialized body, and this specialized body we came to be known in the later time that was the chloroplast. Now further, let's understand the third experiment. The third experiment was explained by the scientist T.W. Engelmann. What did he do? He took the cladophora, which is a green algae, and then he took the suspension of aerobic bacteria. The word aerobic, it means the bacteria which grows in presence of oxygen. Now he took the suspension into this, and then he took the prism and passed the light. Now the observation from this experiment was that the light that had split through the prism and the bacteria that grows abundantly into two spectrum of light that were blue and red light. This blue and red light gave the conclusion that the aerobic bacterial growth was observed maximum. So this was the conclusion from the scientist of T.W. Engelmann. Now, the another experiment was explained by the scientist whose name is Cornelius van Niel. Now he took or he used the purple sulfur and green bacteria. Now as per his experiment, the equation carbon dioxide plus H2S in presence of light, there was formation of glucose and the byproduct was sulfur. But this sulfur came from H2S. This was his experiment. But in case of plant, if we took or if we learn the experiment about the carbon dioxide plus water in presence of light, the reaction was C6H12O6 glucose and byproduct was oxygen. This oxygen comes from the water. This was his conclusion from the previous experiment by using purple sulfur and green sulfur bacteria. Now further, he used the isotope O18 for H2O and O16 for carbon dioxide. Now the result which he obtained was O18 that was found in the oxygen as a byproduct. From that he concluded that the oxygen which is a byproduct comes from the water. Thank you.